Is There we go. Welcome, everybody. This is our first public webinar that is focused on presentations from the trainees in the center. We've organized these series of talks around some loose themes within ATP-Bio, and this first one is all the talks are going to be about cryoprotective agents. During the talks, we'll have three speakers. So during each speaker, if you have a question, you can type it into the chat sort of during and immediately after the presentation. We'll hold all sort of verbal Q&A for all the presenters at the end. So to kick us off, our first speaker is Rasha, who is a postdoc at MGH. She got her PhD from Carleton University, and today she'll be talking about cryoprotectants loading in zebrafish. So Rasha, I'll hand it off to you.
Thank you so much. I appreciate it. And thank you so much for giving me the opportunity to present some of my work here with you today. So let me just get the setup started. So, and thank you guys for joining the session today. So my name is Rasha Al-attar. I'm a postdoctoral research fellow at the Shannon Teste Lab at the Center for Engineering, Medicine, and Surgery at Harvard Medical School and the Massachusetts General Hospital. And today I'll be giving you guys a talk about how we developed this new loading protocol in order to, like, in a non-invasive manner, introduce CPA into the zebrafish embryos. So why zebrafish? So zebrafish are a great research tool because they're easy to maintain, they're cheap, they're amenable to genetic modifications, they're widely studied, and they share with over 70% gene homology with humans. And around the world, there's so many transgenic lines and disease models that have been already made, but we are at risk of losing them, many of these valuable tools, because we don't have adequate preservation strategies for them. Sorry, my bad. So right now, if you want to keep a zebrafish line, you either have to keep a livestock, which can become costly over the years. And if there's any mechanical failure or widespread disease, then you risk losing all these lines. You can also freeze the sperm and then have in vitro fertilization to recreate that transgenic line. However, this is very difficult. It's susceptible to genetic changes, and it would take up to about 8 months to a year if everything goes well to recreate that transgenic fish line. So my project at the Teste Lab is to looking into finding ways to cryopreserve zebrafish embryos or larvae for long-term biobanking to facilitate storage and long-distance transfer. So very briefly, the things that we have to consider in order to have successful cryopreservation of the zebrafish. And this is because cryopreservation of the zebrafish embryo is difficult because of its biology. First, we have the chorion, which is a thin membrane that can protect the embryo as it develops. It is porous and allows the exchange of material from outside environment into the perivitellin space. And then you have the perivitellin space, which is right in here. And that basically contains the fluid, proteins, lipids, and it's one of the two major components that affects freezing, because once it expands due to high water contact, it can rupture, affect the embryo and the blastomere. And then you have the yolk. The yolk, which is like it has high lipid contact and is very susceptible to lipid peroxidation. So this is where the blastomere, the layer on top of the embryo, which gives us the zebrafish eventually, gets their nutrients. So even if the cells do survive the freezing process, but the yolk doesn't, then the embryo will die because it doesn't have a place to anchor itself on and will not have access to nutrients. And then you have the blastoderm, which is the actual embryonic cells that could be damaged with excessive dehydration, oxidative stress, and the freeze-thawing process. So therefore, understanding this would allow us to find a freezing protocol that works basically in an easy way to cryopreserve these zebrafish embryos, so that even labs that don't have the specialty or have the specialized equipment would be able to cryopreserve their cell lines, their zebrafish lines. So the loading of the zebrafish embryos, currently you can either do microinjection or you can submerge them into the CPA cocktail. However, each technique has its pros and cons. With microinjections, you can get you can control the local delivery. However, by injecting the embryo, by injecting the zebrafish embryo, whether it be in the yolk or within the cells, you cause physical damage. It is operator variability, so you do need to have somebody who's trained well enough to be able to do this adequately. You have non-uniform CPA distribution. You're restricted by the volume of the CPA that you have to inject. So you have to inject high concentrations, and because of that, you have very local induced toxicity, and this has very limited scalability. On the other hand, if you're doing submergence, is that you have heterogeneous uptake, but you have limited control over like the CPA concentrations, and it requires like prolonged equilibrations. The cons of it is that it's low risk. Sorry, I switched the pros and cons. I should have checked. The protocol dependent, it is scalable. It causes like moderate damage and then moderate uniformity of CPA distribution. So here at the Tessie Lab, I wanted to create a protocol that merged the benefits of both of these techniques. So what I came up with is that I developed this dehydration cocktail that can basically dehydrate the fluids in the perivitellin space so that the embryo becomes completely dehydrated. So then when we put it into the CPA and it gets rehydrated, you have 100% CPA that's going to be going into the perivitellin space in order to expose the embryo to that. So as you can see here, we have like the various concentrations of my dehydration cocktail, and we have the different times. So as you can see, with the higher concentrations, you get more dehydration over time, like within the 30-minute time point. And as you can see, the fluids in the perivitellin space decreases, the chorion collapses, but the embryo stays intact. Similarly, if you submerge them back into either the embryo media or if you submerge them into the cryoprotective cocktail, you can rehydrate these embryos and there is minimal damage to the actual embryo. And that way you have an easy way of introducing your CPA close to where the embryo is. So this simple protocol allows us to very simply introduce and reproducibly introduce CPAs into the embryo. Is very scalable, it's non-invasive, and it allows us, more importantly, to know the exact concentrations of the CPA that is surrounding the embryo. So unfortunately, right in the middle of the experiment, Fisher decided to discontinue one of the major components of the cocktail. So now, in collaborations with my new colleague, Xiaopei Wang, we are re-optimizing a substitute to basically design the same cocktail with a similar efficiency. So the results that I've showed you in the previous slides are using the substitute that we were doing, but the results that I'm going to show you after this are actually the results that I've generated using the previous cocktail. But our goal is to recreate the results that I'm going to show you with the new substitute. So basically, to take a step back, in order for us to have a successful cryopreservation protocol, we need to have multiple steps optimized. We need to adequately replace the perivitellin space fluid with CPAs. We need to optimize a CPA composition that is not injurious to the actual embryo. We need to have adequate rehydration with the CPA. We need to optimize a freezing protocol. We need to optimize the storage conditions and the storage time. We need to optimize the slow thawing protocol, and we have to remove the CPA, and then only then we can have a successful cryopreservation protocol, and then basically take the embryo into adulthood. So very simply, like in my protocol, I dehydrate. Oh, can you guys still see my slides? Oh, my apologies. Okay, can you see them now?
Yep.
Okay, fantastic. So basically what I do is that I dehydrate the embryos in mass, and then I put them in a petri dish, maybe like 100 or so embryos at a time with my CPA. And then I add my antioxidants, I freeze them, and then I thaw them gradually at colder temperatures, remove the CPA, and watch the development. So some of the preliminary results that I have so what I did, and these are using the old dehydration cocktail, I tested 0.5% dehydration and 1% dehydration for 30 minutes or 24 hours. So I dehydrated the embryos for that time, but I saw that with 1% dehydration cocktail, all the embryos pretty much are dead within 30 minutes. So I stuck with a lower concentration of 0.5% dehydration cocktail. And I saw that there's no difference in terms of dehydration rate if I keep the embryos for 30 minutes or for 24 hours. Keep in mind, dehydrating the embryos for 24 hours does delay their development, but does not impede it, like does not cause any deformities. But because with 30 minutes we've been getting the good enough viability and adequate dehydration, I carried the experiment forward using the 0.5%. And then I let these embryos that have been treated with 0.5% dehydration cocktail develop until 3 days. And as you can see, there is no difference in viability. The heart rate, everything else was the same. So taken from this, I took the 0.5% dehydration cocktail with the 30-minute time point, and then I moved next to the next step to introduce the CPAs into the zebrafish embryos. So previously, I optimized about 6 different cocktails. In here, I have CPA 8, 11, and 12. So what I did here is that I dehydrated the embryos using my dehydration cocktail, and then I rehydrated them with CPA 8, 11, or 12, either at room temperature or at 4 degrees. The reason being is that when I was just submerging the embryos without dehydration, I was getting good enough viability at room temperature, but that was because not the concentration of the CPA within the perivitallin space wasn't 100%. But now that I am introducing 100% CPA into the perivitallin space within where the embryo is, I'm getting loss of viability because of that. So I reduced the temperature of where I introduced the CPA to 4 degrees to reduce the viability. And then I took these embryos, I removed the CPA, added E3 water, and monitored them for 72 hours. And we can see that the viability was more or less the same. The hatching rate is the same between the different CPAs, even though CPA 8 was like a little bit higher. And the heart rate is more or less similar. So from this, I optimized that loading the embryos with CPA has to be done at 4 degrees for maximal viability. And then I went to actually freezing the embryo. So previously, I optimized a slow embryo freezing protocol inspired by the freeze-tolerant wood frogs in nature. So in here, what I did is that I took the embryos, I dehydrated them, I rehydrated them in my CPA 8, and then I did slow freezing, and then I froze them at -8, -15 degrees Celsius for about 24 hours. And in here, you can see that frozen embryos actually look opaque, dark. And then but once you saw them, the dead embryos remain opaque, but then the live embryos become translucent. So even though I had loss in viability, I was still able to get some viable embryos. However, within 3 hours after post-thaw, the cells die because of excessive osmotic stress. So right now we're in the process of re-optimizing the cocktail with a new substitute, and we will be testing different antioxidants and different membrane stabilizers in order to reduce the osmotic stress on these embryos and have them left for longer periods of time. So the take-home message is that the D1 method enables for simple non-invasive loading and unloading of the CPAs in the zebrafish embryo. The CPA rehydration at lower temperatures improves the embryo viability. Gradual removal of CPA is essential to minimize osmotic shock before freezing and after thawing. And CPA unloading should be paired with reintroduction of various antioxidants and membrane stabilizers in order to prevent injury. So this was my presentation, and I'd like to thank Dr. Shannon Tessie for giving me this opportunity, my colleagues, as well as Jape Wang, who's now helping to optimize the various steps with the new substitute. This research was funded partly by the Canadian Institute of Health Research, Postdoctoral Fellowship, and ATP-Bio. Thank you so much, and I'm happy to take questions after the meeting. Thank you.
Thank you, Rasha. If anyone has a quick question that they just want to type into the chat, you can go ahead and do that. I'm sure Rasha will take a look at it. Otherwise, if you have a other question, you can jot it down and save it, and we'll circle back to Q&A for all 3 presenters at the end. With that said, we will move on to our second presentation. Our second speaker is Natalie, who is a current postdoc at MGH as well. She got her PhD from Johns Hopkins, and she'll be talking about CPAs and immune cells. So Natalie, I'll pass it off to you.
Thank you. Hi. Thank you for the introduction. My name is Natalie. I am a postdoctoral fellow in Dr. Mehmet Toner's lab, but I also work very closely with Dr. Rebecca Sandlin, who is overseeing this project. So, Primarily, I'm going to talk about our microfluidic chip that is for high throughput loading of cryoprotective agents into therapeutic immune cells. Immune cell therapies, after the success of CAR T cells, has really expanded and expanded into other cell types. So you can see here, let me turn on my laser pointer. Here in 2024, CAR T cell therapies actually only make up about half of all the immune cell or immunotherapies, cell-based therapies that are in clinical trials right now. And one reason for that is because if we use innate immune cells rather than adaptive immune cells, we can create a more universal therapy because these do not have to be MHC matched. They do not rely on antigen recognition. So they can be kind of made into an off-the-shelf therapy. And why that's important to cryobiologists is that T cells are actually quite robust. They can withstand pretty well the typical like 5 or 10% DMSO loading. But these other innate immune cells don't fare as well. There's a lot of cell death and also loss of function. And this is kind of that typical timeline for what the cryopreservation of either an autologous or allogeneic therapy looks like. So cells are collected either from a healthy donor or the patient itself. These cells are then frozen so that they can be shipped to a manufacturing facility. All cell therapies require these specialized manufacturing facilities, and there's only a handful in the country. So in manufacturing, they are thawed. They usually isolate the cell of interest, potentially do some engineering, and then activate the cells, expand them, do quality control, and then refreeze them for a second time so they can be shipped back to the hospital for the clinical application. So this is not one, but two freeze thaw cycles for these cell products, which is can be quite damaging, especially for a cell type that's not as robust as T cells that can't really handle these processing procedures. This is a table from 2024 that summarizes approved immune cell therapy products that are currently used in the clinic. You can see the majority of them are CAR T cells. And if you look over here at the cryomedia that's used with them, it's almost exclusively DMSO, between 5 and 7.5% DMSO. It's commonly used across the board. There is some reduction in viability, dampened proliferation, a reduction in the function of these cells. But like I said, for T cells, it hasn't been a huge barrier for their use in the clinic. But for all these other cell types that we're now looking at preclinically, um, we need a more robust system in order to load cryoprotective agents and protect them so we can actually use them in patients. So what we're focused on is loading non-toxic CPAs. In particular, we're focusing on trehalose. The problem here being that trehalose is also non-permeable. So there's been a lot of methods that are looked into to get trehalose loaded intracellularly, from single cell methods like microinjection to physical methods like ultrasound or thermal methods, and biomaterials or nanoparticle-based methods as well. But the problem with all of these, even if they do get a clinically relevant amount of CPA into the cell, there's a huge manufacturing burden. For example, microinjection is obviously on a per cell basis. Liposomes and nanoparticles would require a second QC because those biomaterials themselves have to be QC'd. The physical methods often result in really low cell viability afterwards. And then also with a lot of these different methods to load trehalose, there's low throughput as well. So we are interested in using this microfluidic chip that was developed previously in our lab for viscoelastic mechanoporation that we term veloporation. This is what the chip looks like. This is just a normal glass slide for reference, so it's quite small. There are two inlets here and here. The cells go along these spirals. The purpose of the spirals is to get them in a single file line so that once they get to this narrowing chamber or aperture, they go through single file one at a time. So because we have a viscoelastic fluid inside of this chip, once the cells get to this really rapid narrowing point, the forces that the cell experiences looks like this. So it pushes the cell in one direction and stretches the cell in another direction. And because of this stretching, it creates pores in the membrane so that any cargo that's available in this loading media can enter the cell via just diffusion. Um, in our first paper on this, we did this in jerkat cells and loaded CRISPR complexes and also mRNA. And this is a picture of what it actually looks like under the microscope because you see they're quite elongated and these are typically rounds. This is what the cells look like after loading unstretched and then stretched cells. So after that first paper, we've expanded the cell types that we validated. So we've done several different cell lines, both adherent cell lines and suspension cell lines, and then also primary cells. So we've gone into primary T cells, NK cells, neutrophils, hematopoietic stem cells, and also red blood cells. But these first kind of validation in each of these newer cell types was done with just a small fluorescent dextrin. And so now we're adapting the device to be used to actually load trehalose. And I wanted to briefly talk about the different parameters that we're changing in order to adapt these cells to loading. So we're trying to change basically how much force the cell experiences within this narrowing aperture here. So if we're really gentle on the cells, we'll have high viability, but we won't have as much stretching of that cell, which means we'll have less poration and less loading. If we rather increase the forces that the cell is experiencing, we'll get more stretching, more poration, more loading. But if we take it too far, we'll get cell lysis or just initiation of apoptosis. So how we change the forces that that cell experiences in this chamber is the length of the narrow chamber, the width of the chamber, the polymer type that we're using as that viscoelastic fluid, the length of the polymer concentration, and also the flow rate at which the cells are being pushed through the chip. So again, this is what that looks like. So when we're loading the cells into the microfluidic chip, we have the cells, we have the cargo itself, in this case, trehalose, and then we have our viscoelastic polymer. And first, we just took jerkats since we know they work really well in this chip and tested different kinds of viscoelastic polymers to see which one works best with loading trehalose. So we looked at PEO, which is polyethylene oxide, is just a long version of PEG, and also hyaluronic acid. And we saw across the board, we have about the same viability depending on what polymer we use and what size of that polymer we use. But when we look at delivery, we have a pretty drastic differences. So first off, once we put trehalose, and in this case, we're using 0.2 molar trehalose, in all cases, we do see a reduction in the amount of loading of cells that we're getting. And again, I'm using loading kind of loosely here. We're using a proxy fluorescent molecule to determine trehalose loading. And I'll get into actual trehalose measurements later. So we do see a reduction whenever we put in 0.2 molar trehalose, but we did see when we use this 2 megadalton PEO molecule, we're getting quite high delivery, both with the trehalose and without the trehalose. So all the experiments moving forward are with this 2 megadalton PEO. So that was with 0.2 molar. We wanted to see how high we can take this. So we used jerkat cells with the parameters on the previous slide and tested 0 molar all the way to 1 molar. And as you can see, we can get up to about 0.5 molar without a huge loss in viability and loading. It's about 80% viability, 80% loading. Once we go to 1 molar, we can load all of the cells, but they are dead. So we're working on kind of looking at where exactly that cutoff point is. But right now we can get at least a 0.5 molar, which is really clinically relevant. A lot of the literature looking at trehalose loading into cells is around this range, about the 0.1 to 0.2 molar. So we can definitely get that in our kind of test cells. So once we saw this, we moved on to our cell that we're interested in. And first, we're trying to expand this to a lot of different cells. But first, we're looking at neutrophils because neutrophils are notoriously hard to work with. They die very quickly. If you want to use them clinically, you have to use them right away after they're isolated. Or we need to find a way to cryoprotect them because right now there is no really viable method to cryoprotect neutrophils. So that's why we chose neutrophils. And on this slide, all these experiments were done with zero point 0.2 molar trehalase. First, and we haven't used neutrophils in this microfluidic chip before, so that's why we had to start at the beginning and kind of look at all of the different parameters to find that sweet spot of high loading and high viability. So first we look at the stretching channel dimensions, which again is the shape and size of this narrowed part of the chip here. Then we looked at the pressure of the flow, so how much pressure is used to push the cells through the chip, and then also the polymer concentration, using that same polymer that we saw worked really well in Jurkat cells. So we started to get pretty good results here at 90% viability, 75% loading. But again, this is all done, when I say loading, it's done by co-loading a small fluorescent dextrin kind of as a proxy to determine how much is actually getting into the cells. So we wanted to look at a more concrete measurement of is trehalose itself actually getting into the cells. So we loaded cells using various concentrations of trehalose in the loading solution. So just that's the concentration of trehalose outside of the cell when it's porated. We washed them several times and then pelleted the cells and gave them over to the mass spec core at Harvard so they can lyse them and measure intracellular trehalose. And what was really exciting is that we do see as we increase the concentration of trehalose in that loading solution, we are getting higher intracellular trehalose. And it's almost at some points equilibrating between the outside trehalose concentration and the inside. We do, as we increase that trehalose concentration on the outside, we do start to plateau of how much trehalose we're getting onto the inside. But we are going to repeat this just to make sure we're getting the same trend and also the same trend in different cell types. But this makes sense to us because we know that when we look at how long it takes for the pores that form in the chamber to close, the really large pores close quite quickly within 5 seconds. But the smaller pores are open for, up to a minute, which makes sense in terms of the timeline for how fast this diffusion can happen. So it's following the principles that we expected. And finally, we did functional testing on these neutrophils just to make sure that although they have a high viability after loading, are they actually still clinically viable? So again, at our best, we're getting about, and I'm here comparing cells that went through the chip but without trehalose, and then went through the chip with trehalose. And then control is just freshly isolated cells. So at our best, we're getting about 75% of the cells loaded with zero point 0.2 molar trehalose, about 96% viability. And then when we look at phagocytosis as a function or as an assay to assess function of the neutrophils, we get pretty comparable phagocytosis compared to freshly isolated cells. And then when we look at surface markers as kind of a proxy for cell activation, it's also comparable. So as you can see, we get a little bit more phagocytosis and a little bit higher expression of these surface markers once the cells have been put through the chip. But not a huge amount. So it seems like they are getting activated somewhat, but not drastically to where it might be a problem clinically. But we are going to continue these functional studies and compare it to the clinical standard of 5 to 10% DMSO to make sure these cells are viable moving forward. We're also going to extend these studies to other cell types. We'd love to work with collaborators if anyone is interested. And I see kind of long term this being kind of a one-pot system for engineering and cryopreserving these cells at the same time. So taking donor cells, co-loading both the cryoprotectant and the cargo of choice, freezing them down, and then these become kind of universal off-the-shelf cells that can then just in one step be thawed and expanded at the hospital and then reinfused. But that's, looking ahead into the future. So I'd like to thank my advisors, Dr. Toner and Dr. Sandlin, and everyone in the lab that's been a huge help. And also want to thank Dr. Mishra and Dr. Ramia, who have also provided their guidance, and of course our funding sources and ATP-Bio. All right, and happy to take questions in the chat. Thank you.
Thank you, Natalie. Again, if you have quick questions, you can write them in the chat and Natalie will get to them. And again, we'll save sort of longer form questions for the end for all of the presenters. Our last speaker today is Zhang Hu, who is a postdoc at the University of Minnesota. He also completed his PhD at the University of Minnesota. And today he'll talk about organ CPA perfusion. So Zhang Hu, I'll pass it to you.
Thank you. Hello. Hi. Can everybody see my screen fine?
Yep, looks good.
Okay, perfect. Yep. So today my topic is improving organ CPA perfusion via a multi-thermic strategy. So as we all know that the organ shortage is huge and there are more than 100,000 people on the waiting list every year. And the transplant surgery done every year is only like about 40,000 every year. However, there are still a huge amount of organs are discarded every year. And for example, in 2025, there are more than 11,000 organs are discarded. So to clarify that, those organs are recovered, but they have to be discarded due to this or that problem. And the main problem is because of the preservation time. So therefore, if we can extend the preservation time, we can save a lot of organs. So in ATP-Bio, we focus on a few technologies to extend that preservation time. We have hypothermia, supercooling, partial freezing, and those are on the both of them are on the high sub-zero temperature range. And our lab focused on the vitrification, which is the low sub-zero temperature range. And technically, the vitrification can extend the storage time to indefinite. So that will solve the problem for wasting organs. And here is a brief history of organ vitrification. So back in the 1984, Dr. Greg Fahey achieved the first mammalian organ vitrification. And this is the famous photo of comparison between the frozen and the vitrification picture. And however, researchers were not able to rewarm those vitrified organs back to normal temperature without any ice formation. And after 25 years, in 2009, they were able to achieve a single success of successful vitrification and convective rewarming. However, due to the convective warming is a slow and non-uniform rewarming technique, so that success was not quite reproducible. Therefore, later in 2014, my professor, Professor John Bischoff, our lab introduced the nanowarming technology, where we perfused the nanoparticles to the organs inside and put that under a radio frequency coil. And those particles will generate heat, uniform and fast heat to rewarm the vitrified organs. And after that, our lab showed a few physical proof of principles of nanowarming in multiple organs, including kidney, liver, and hearts. However, we were struggling about the toxicity issue. Therefore, I developed some modeling to minimize the toxicity while perfusing into the organ, perfusing CPA into the organ. And we have achieved some success. And eventually, we were able to achieve some reproducible success of vitrification, nanowarming, and transplant success. And most recently, that was in rat kidney as well. And most recently, Dr. Woke and Dr. Fahey, their group were able to scale the success from rat to rabbit kidneys, and they were able to achieve some reproducible transplant success as well using vitrification and dielectric rewarming. So vitrification, many of us are very familiar. Vitrification is a ice-free cryopreservation technology where the sample is cooled fast to avoid ice to enter this glass-like amorphous state. And for different scales of samples, we use different concentrations of CPAs and we achieve different because we can achieve different scales of cooling rates. And here is a relationship to show that critical cooling rate dependency on the CPA concentration. So the more concentration of CPA we have, the lower critical cooling rate is needed. And for organs, especially, we were only able to achieve a slow cooling rate in the core of the organ. Therefore, we will need a high concentration of CPAs. As I mentioned, those success were achieved in the rodents, either rat or rabbit kidneys. So therefore, if we want to achieve human-sized kidneys or human-sized organs in general, vitrification, what kind of cooling rate is achievable and what kind of CPA is needed? And here is a figure to show that's achievable cooling rate, which is recently published in Nature.com by Rasha. So as we can see, for different size of organs, for kidney is the smallest for organs, and we can achieve approximately 2 C per minute in the center of the organ. And for larger organs like heart, lungs, and liver, we can only achieve less than 2, even less than 1 C per minute, especially for the liver. Therefore, if we want to focus on the kidney, we can achieve 2 C per minute cooling rate, and we substitute that cooling rate back to the relationship between CCR and the concentration. We can get approximately like 60%-ish weight by volume concentration. And that refers to the CPA concentration that we need to load to the CPA before vitrification to achieve successful vitrification. And 60% CPA concentration is approximately 8 to 9 molar CPA concentration, molarity, depending on the CPA compositions. However, as we all know, that high of a CPA concentration is quite toxic. So in the left figure, we can see that the toxicity rate dependency on the CPA concentration. So the solid curve is the toxicity rate for VMP, and the dotted curves are for some more concentrated CPAs, but also in the VMP family, so VM3 and M22. So we can clearly see that the toxicity rate increased really drastically in this CPA concentration range between 8 and 9 molar. Therefore, the toxicity is also like harming the organ during perfusion really, really, really bad. And to reduce that much of toxicity, a common way to do that is to lower the perfusion temperature. For example, Dr. Fahey has proposed using a minus 22 Celsius to load the CPA M22 for the kidneys. So that corresponds to this CPA concentration. Both full strength VMP and both full strength M22 are perfused at negative 22 Celsius. But there isn't a detailed study on why we're choosing that temperature or the kinetics of choosing or the reasons of choosing that temperature. Because as we know, when we are reducing the CPA temperature, we're not only reducing the toxicity rate, but also we're reducing the permeability, which can potentially extend that's CPA exposure time and increasing the CPA, the accumulative CPA toxicity. Therefore, on the one hand, we're reducing the toxicity rate, but on the other hand, we are extending CPA exposure time. Therefore, there's a balance between those two, and there should be a optimal temperature for that kind of problem. So in the left figure, we can see that the temperature dependency of the toxicity rate, and the toxicity rate temperature dependency is assumed to be a Arrhenius relationship. And on the right side, this is very well established. So the permeability is also a parameter of the temperature and following the Arrhenius equations. And if we look into those equations, they're quite similar, almost exactly the same, except for this C here, concentration. Therefore, ideally, theoretically, there's for a certain CPA temperature, CPA concentration here, there should be an ideal perfusing temperature to minimize the toxicity while ensuring adequate CPA perfusion. Therefore, we did a really quick, simple case study to kind of illustrate this multithermic perfusion protocol that we proposed. So for both monothermic, which means just one temperature, and multithermic perfusion protocols, we have the same target intracellular CPA concentration, 8.5 molar. And for the monothermic perfusion, we use one single ramp rate, which is 50 millimolar per minute, which is the gold standard for the rat and rabbit kidney perfusions. And for the multithermic, we use different ramp rates, 100 to start with at the lower CPA concentration, and 50 at the intermediate CPA concentration, and a slower ramp rate, 35, at the high CPA concentrations. And for the temperatures, for the monothermic perfusion, we followed the rat and rabbit kidney loading protocol, which is where we set the temperature to 4 Celsius. But for the multithermic perfusion, for the lower CP concentrations, we have a higher temperature to facilitate that permeability CPA loading. And for the intermediate CPA concentration, we still have 4 Celsius. But for a higher CPA concentration, we reduced that perfusion temperature to reduce the CPA toxicity. And both of the protocols will increase to a final concentration of 9 molar and keep it there until the intracellular CPA concentration reach 8.5 molar. And the total time calculated by using those two protocols are quite similar. It's about 190 minutes. And those first two figures show that the arterial CPA concentration, which is the loading CPA concentration, represented by the black curves, and the tissue concentration, which are the intracellular CPA concentration inside the cells, which represented by the red or blue curves. So, and for the figure C, that is the calculated accumulative toxicity inside the tissue for both protocols. So for the normal monothermic perfusion protocol, we have larger than 0.3 J-tox. J-tox is the toxicity cost function, which represents the accumulative toxicity inside the cells. And based on these J-tox values, we can easily calculate the predicted viability. And the larger J-tox indicates a larger toxicity and a lower viability. And we can see that the monothermic perfusion almost created like more than twice of the toxicity accumulated inside the organ. And the predicted viability from those two protocols are 70 versus 85% after the CPA loading. So that difference was purely due to the difference protocols. And toxicity rate, toxicity effect is one side, but the other side is the osmotic damage. And we also looked into the osmotic damage, which is the volume. So during CPA perfusion, the volume always shrinks, and we want to check whether the osmotic stress is similar, and the volume represented is the volume change is represented here, and we can see that the osmotic stress is also similar. Therefore, multithermic has a great advantage compared to monothermic loading. And here are a few conclusions we have. So for multithermic perfusion, we can enhance the perfusion efficiency at the lower temperature, lower CPA concentration by increasing the temperature and facilitate the CPA permeation into the cells. It can also reduce the CPA toxicity at higher concentration by decreasing the temperature and the ramp rate to kind of minimize that toxicity. And as I mentioned, that is only a very preliminary case study on the multithermic perfusion protocols, and a more detailed model may be needed to explore the real potential of that multithermic perfusion. And I would like to acknowledge my lab, Dr. John Bischoff and Eric Finger, and all the collaborators. Thank you.
Thank you, Zhang-Hu. So that concludes our 3 rounds of speakers. Be keeping an eye on the chat here, and I think all the chat questions have been answered. So if anyone has a question that they'd like to unmute and ask, now is the time for that. If you're in a spot where you can't ask verbally, we can kind of read it from the chat. But we can go ahead. I guess we can do the raise hand function just to keep it orderly.
I have a
Yeah, please go for it.
Sure. Rasha, thank you. Thank you, everybody. Those were all great talks. Really appreciate it. Yeah, so you answered me in the chat about your working for up to like maybe a week or so for -15, and then you wanted to go longer, potentially later. Do you think that there's a way, and then I also saw you were answering Laksha about, the ice being in the paravital space. Do you think that there's a way to confine the ice so that it won't be damaging and that you could potentially go all the way down? Is that kind of the idea overall?
So that is the idea overall. So what we're trying to do is that we want to stabilize our stabilize our results at like -15, and then kind of like dunk them in liquid nitrogen thereafter to allow them to go down to to deeper cryogenic temperatures. So the goal is to do that, because whenever we try to do slow freezing using our CPA cocktail down to like -40, like you can't even distinguish between the blastomere or the yolk after we thaw them. So it's definitely some sort of ice damage that is happening. The sweet spot that we found is -15 degrees Celsius. And right now we do have to modify a few things in our CPA in order to be able to go to deeper cryogenic temperatures. But, with the new substitute and us having to redo all the experiments, again, I think it's going to be a few months ahead. But the goal is to try to restrict the ice even further and then go to deeper cryogenic temperatures.
So, I mean, just a quick follow-up on that.
Yeah.
Your rates will become important, I think, because
Yes.
you're going to create. I hope you hope that you're going to be creating a vitrified, middle.
Yeah.
And
So
the
that
rates
is
will become
that
important.
is the goal. We've tried a couple of experiments just, of hands to see like if it works or not. And so far we haven't had any success yet. The one thing is that with the slow freezing protocol that I'm doing right now, our CPA concentrations are extremely low. Versus for vitrification, you want to have really high like CPA concentrations. So Is this the reason why we're not getting any viability whenever we do vitrification? I don't know. The issue right now is that if we cannot successfully thaw and develop the embryo to adulthood from -15, we're not even anywhere close into trying vitrification because even if we have successful vitrification and rewarming, but the embryos are dying at temperatures, below -15, then we can't really distinguish where the damage is happening.
That's a very good point. Thank you, Rasha.
No problem. Thank you for a question.
All right, I see Adam, you have your hand up. You can go ahead and ask a question.
Sure, yeah, my question is for Zonghu. I thought your presentation was really interesting, and I didn't catch what you were using as your transport model. Are you using an organ model for that or a cell permeability model?
Yes, sorry about that. I forgot to mention. It's the organ model. It's the Crow cylinder model. My apologies.
Okay, cool.
Brian, you can go ahead.
Yeah, I also enjoyed your talk, Zhanghu. My question is, given that you have a complete mathematical model you're using for toxicity and permeation as a function of temperature, could you use the calculus of variations or some other mathematical model to derive a continuous variation in temperature as a function of concentration to reach like the absolute minimum in toxicity instead of step functions, or is the toxicity actually minimized by step functions of temperature?
Very good point. Yes, I totally agree. But limited by my math skills, I myself I'm not quite able to, yeah, achieve that. Optimal is definitely not optimal. Just a quick case study to show the benefits of multithermic protocols. But there should be, yes, as you mentioned, there should be a continuous change ramp rate and temperature, ideally. Yes, yes, yeah.
I don't see a name attached to it. I just see iPhone. So whoever's joining via phone, you can go ahead and ask.
I'm sorry about that. Yes, I guess I am iPhone. Um, um, my question is for Rasha and also just to all the presenters. Fantastic presentations, really, really just incredible, mind-bending stuff. Um, I'm sorry if this was already asked, but have you done anything explicitly to stabilize the lipids in the yolk, or does that just kind of work out?
No, it does not work out. And it did take me a couple of months to do it. So actually that's that's one of the very first things that I had to optimize after CPA loading because the yolk is the first thing that gets damaged, whether it be freezing or just even introducing the CPA. So
Yeah.
I have to actually use a lot of antioxidants, different
Mm.
combinations of them as well, and various concentrations in order to stabilize the yolk so that I prevent that lipid peroxidation from happening.
Okay, so it's antioxidants you found like stabilize membranes and lipids?
Yes. Yeah.
Awesome. Awesome. That's super interesting and useful for me to know. So thank you for that.
Good. Happy to chat offline if you're interested.
Of course. Yeah. Thank you.
Yeah. Mhm.
I guess I have another question for Natalie. Thank you. That was a very nice talk. It seems like that platform is so flexible. How do you choose which next cells you're going to work on? I know funding probably plays a role, but what are you thinking in terms of if it was up to you and Mehmet, how would you prioritize the cells that you want to work on?
Yeah, funding and collaborators, definitely. But we're also working on naive T cells. So a lot of work in loading T cells is done when they're already activated. And that is easier because they're larger cells once they're activated, and they also uptake molecules more readily. So I think there is a break in the market for loading fully naive T cells. So I think that's something that we're interested in. Yeah, but I think just looking at, market gaps and what has been tricky to load in the past.
And then is your strategy once loaded, are you trying both slow freezing, vitrification, and all the above, or are you? What do you?
All the above, all the above. Yeah, I'll say that now while it's recording.
Okay. Thank you. Very nice work.
Thank you.
Charles, why don't you go ahead?
Hey, Zhonghu, I'm new to this JTOX variable. Are you gonna try to show this like empirically that there's this reduction in toxicity with your perfusion settings, or have you already done that?
That's a really good question. Yes.
Okay.
Yeah.
Cool.
Well, why don't you talk
Thank you.
about how you got it?
Yeah, the model.
No, the JTOX.
Oh, JTOX. Yeah, JTOX is based on Dr. Higgins and Dr. Benson's toxicity cost rate function, toxicity cost function. So pretty much is every CPA, when the cells are exposed in the CPAs, the CPA inside the cell is killing the cells every moment, and different concentration of CPAs are killing the cell at different rates, and the toxicity rate is based on that. So pretty much it's just a rate number, and over the time it will kill the cell eventually. And to calculate this accumulative toxicity effect, they come up with this toxicity cost function to calculate the entire number called JTOX, the toxicity cost function. And using this JTOX, we can easily calculate the viability, the predicted viability after a certain amount of exposure time in CPA. And it's supposed to be a exponential plugin. It's a very easy. N divided by N0 equals exponential times that JTOX minus JTOX. Yeah. I'm pretty sure Dr. Higgins can share.
Yeah, how do you do, Adam?
I'm wrong. Okay, thank you. Yes, I did it right. Yes.
Thank you.
I think we have time for maybe one more question if anyone has any a lingering question that they have not asked yet.
I have a question.
Please
Who was the fella on the iPhone? At first I thought it was Nick Zuckowitz, but then I'm just like, nah, that doesn't sound like Nick. Is he still on?
The iPhone's still here.
Mr.
It sounded like
iPhone.
maybe Mark Woodward.
Oh, was that Mark? Yeah, that did sound a little like Mark.
No, I'm on a separate. I think I
Oh,
labeled
you
myself
are.
off
Hey,
on
Mark.
the-
Hi
Hey.
there. Yeah.
Forever be a mystery.
Well, I have one more question. What does the collected audience here think about having webinars where our trainees give multiple talks like this? Was this a success? People think so? Yeah. Cool.
All right, well, the good news, we'll have it again. Off the top of my head, I do not know the theme, or I think the date might be in April. So keep a lookout on your emails for the next trainee talk. But until then, the next webinar is going to be the State of the Center, and that'll be in 2 weeks on February 17th. There'll be an email with information there, so be on the lookout for that. Other than that, thanks for joining, everyone. Thanks to our 3 speakers, and have a great rest of the afternoon.
Thanks, Jennifer. Thanks, everybody. Thanks.